Hello. Please hit like button and subscribe my channel. Also press bell icon for future video notifications. Thanks.
Imagine using stem cells from your bone marrow to grow a piece of bone tissue in the lab, after which medical doctors explore which drugs have the desired effect on your bones. In this way, a tailor-made treatment plan would be made for everyone, with the best approach being clear in advance. Personalized medicine at its best. That vision of the future is no longer science fiction now that researchers from Eindhoven University of Technology and Radboud University Medical Center have actually realized the first part, growing a lifelike piece of bone tissue from human stem cells. It is the first organoid of bone, a simplified version of the original, the researchers report today in the journal Advanced Functional Materials. Coherent picture. With this, we present, for the first time, the full picture of early-stage bone formation, says Sandra Hofmann, associate professor in Bioengineering Bone from 2E. And that is of great importance. How our bones are formed is still largely a mystery. Bone is a very complex material in which, on the one hand, countless cells and processes interact and, on the other hand, an ingenious matrix of collagen and mineral is built up to provide strength. Much is known about the individual components, but a coherent picture has been lacking until now. Three types of cells play the main role in bone formation: osteoblasts, which build bone tissue; osteoclasts, which take bone away; and osteocytes, which regulate the building and breaking down of bone. Most studies so far have focused on one of these types of cells, but that is not a good representation of the real tissue, says Hofmann. We present here a piece of woven bone, early stage bone that developed from stem cells and contains two types of these cells, osteoblasts and osteocytes. We now see that we can make lifelike bone exclusively with these two cell types, getting wiser from molecular poking, and perhaps more importantly, our system behaves just like early-stage bone, says Anat Akiva, assistant professor cell biology at Radboud MC. We show that both types of cells produce the proteins that they need for their functionality, and we show with the greatest detail that the matrix actually is the bone matrix we see in real tissue. The fact that a simplified representation of the formation of bone at the molecular level is now p. A bone consists of 99% collagen and minerals, but there is also another 1% of proteins that are essential for successful bone formation, explains Professor Nico Sommerdiek from Radboud MC. So what's the role of these proteins? How do they support bone formation? Never before have we been able to look at the milestones of this process at a molecular level. And with that, they immediately have a good entrance to investigate the cause of genetic bone diseases such as brittle bone disease and their possible treatments. Remember that the origin of many diseases is at the molecular level, and so is the treatment, says Akiva. In fact, we now have a simple system in a reliable environment in which we can poke around and see how bone cells react to the stimuli we provide. Story source: materials provided by Eindhoven University of Technology. Note: content may be edited for style and length. Please support my channel to grow by pressing subscribe button and the bell icon. We will notify you technological news. Thank you.